Wednesday, we left off talking about genetic variation in a population and different ways that we could calculate this, right? We went through calculations of how to look at allele frequencies, how to look at genotypic frequencies. We also looked at the Hardy-Weinberg equation. We're going to start today's class by diving into a practice problem on the Hardy-Weinberg equation, right? So my example problem, and this is going to be very similar to an example problem you could see show up on the exam. I guarantee you there's going to be some sort of a sample question about Hardy-Weinberg on the exam. But I might tell you, in a population of deer, right, the population is 200, 65 of them are white, which is the recessive allele, right? Just based on that information alone, I want you to calculate the allele frequencies and the genotype frequencies, right? This is doable. It seems like I'm not giving you a lot of information, but I am. I'm giving you way more than you think. All right, so we have our deer population here. 65 of them are white. The recessive allele is the white allele. So what do we know? All right. Well, if we were to look at this population, we should be able to tell what the genotype is of the white deer. Visiting? Yes. Yes. Go ahead and find a seat wherever there is open here. So we know that 65 of them are white. If the deer is white, right, what is the genotype of this deer? Right, it has the recessive allele. They both have to be lowercase, right? Because if there's any instance of having a brown deer versus the white deer, right, the dominant allele would be masking the recessive allele, right? So we know the genotype of 65 of these deer, right? It has to be little a, or homozygous recessive, right? So we know this. We can also speculate what the genotype of the brown deer is. What is the potential genotype for this brown deer? Yes, I'm hearing all the correct answers. It could be homozygous dominant, or it could be heterozygous. Right? It just needs one of those dominant alleles. Now, it might have both alleles being dominant, and only one allele being dominant if it's heterozygote. We don't necessarily know. We just know it's one of those two possibilities. All right, so we know we have a population of 200 deer. We know 65 of them are white. 65 of them has to be homozygous recessive, right? We also know our equilibrium equations, all right? We know P plus Q equals 1. We know P squared plus 2PQ plus Q squared equals 1. All right, so we know that the first equation, that's the allele frequency. P and Q, respectively, are different alleles. And we know that the second equation is the genotype frequency. All right, we could assign those two alleles to our letters. All right, we could say that P equals our dominant allele of capital A. We could say that Q equals our lowercase a, or our recessive allele. Right? Now, it really doesn't matter which letter you assign to which allele. I tend to do, since P comes first in our equation, I tend to do the dominant allele, because if you write out a genotype, typically the dominant allele is the one that comes first. That's how it works in my mind. You could do this oppositely, and you would get the same answers, right? So we're going to say P equals capital A, Q equals lowercase a. And again, we know 65 of the 200 deer are white. Right? We can really easily calculate out the genotypic frequency of being homozygous recessive. All we have to do is say 65 divided by the total population of 200, and we get this 0.33. Or if we times it by 100, we would say 33%. Right? So we already know this information. Yes. So 33% of them are homozygous recessive. Right? We already know part of this equation of our Hardy-Weinberg equilibrium. pausing to make sure everybody can get their notes down. All right, so we're going to take what we know about this population. And again, we were only given that starting information that we have 65 white deer in a population of 200. We've dissected that information out, and we've gotten all of this information as a result. All right, so the genotypic frequency of lowercase a, homozygous recessive, is 33%. All right, there's going to be a lot of text on the next few slides. I'm going to rely on the slides for some of that note-taking, but all the equation stuff that we are going to do, I will do in this green square. Right, but I've put like reminders up at the top. Reminder, what is the equilibrium? What are our P's and Q's equaling? So we're going to take what we already know. We already know that 33% of the deer are white. Right? We know we have our allele frequency, P plus Q equals 1, which we have said is capital A plus lowercase a equals 1. We already know what our genotype frequency is, which is homozygous dominant squared plus 2 times heterozygous plus homozygous recessive squared equals 1. Now, out of these equations, we already know what the frequency of homozygous recessive is. Right? We've calculated that. We know it's 33%, or 0.33. Oh, my captions just went away on me. All right. While I refresh this page, and hopefully it connects, right? And you can see it as it's trying to connect. We could plug in that 0.33 for our Q squared, right? It would be 0.33 squared, right? But that 0.33, we would take the square root to find an instance of just one of those single A's. Because 0.33 is telling us the percentage of having 2 A's in our genotype at a time. Right? There it goes. Internet must have just cut out for a little bit, is what I'm suspecting happened. All right, so 0.33 represents both A's. And in our equation for genotype, it's squared. So in order to find the frequency of just a single A, we have to take the square root of 0.33. And when we do that, we get 0.57. So now we know A equals 0.57. Lowercase a equals 0.57. The frequency of having the recessive allele in the population is 57%. Right? So we have found the frequency of one of our alleles already. There we go. Yeah?
So is 0.33 squared or is 0.33 equal to Q squared?
0.33 is equal to Q squared.
Okay.
Yep.
So
Yep. Q would be a single A. Q squared is the genotype. And we know the genotype is 0.33% of recessive, homozygous recessive, which is Q squared. Yep. Yep. No, that's a common point of stickiness when students do this equation, right? In the genotype frequency, the squared is linked to the Q at this point. We have to unlink it, which is where we divide it or take the square root of it in order to get 0.57. All right, then we have the frequency of just a single A. All right, so we know as A is 0.57, right? We also know we have this frequency equation for our alleles. P plus Q equals 1. Or capital A plus lowercase a equals 1. Well, if we know what lowercase a is, we can plug that into the equation. Right? So we know P plus 0.57 equals 1. We could rearrange the equation to go 1 minus 0.57. Right? And the only possible answer is 0.43. In order for P plus 0.57 equals 1, P has to equal 0.43. And again, we know P, we have assigned the dominant allele. So capital A, the frequency of the dominant allele, is 0.43, or 43%. All right, so we know the frequency of alleles already. Right? We know capital A is 0.43%, lowercase a is 57%. Excuse me, I should say is 43%, not 0.43%. All right. Now we just have to plug in the numbers, right? We know what A is. We know what capital A is. We know what lowercase a is. We know that they apply to P and Q. We just have to plug our numbers into our genotypic frequency equation, right? So 0.43 squared for a homozygous dominant times 2 times 0.43 times 0.47, one of each of our alleles, plus 0.57 squared. If we were to do the math, we should get these numbers, which again should equal 1. And we know that relates to the specific genotype. Right, so we answered our original question. Right? We know the percentage of deer that are homozygous dominant is 19%. We know the percentage of deer that are heterozygous is 50%. And we know the percentage of deer that are homozygous recessive is 33%, right? Which we already kind of knew from the get-go. Now, if you are actually adding up these numbers, I think you get 1.02. Yeah? And that is still okay, because what has happened here is we have rounded our numbers. So when we add it up, we get a number from the rounded numbers. If we had taken all of these numbers and put it out to like 3 decimal points, you'd probably get something more similar to just 1. But because we rounded, and the way we rounded, it's just slightly off, right? But it's still very, very close to 1. All right, so we have now answered our questions. Oh, my computer is about to die. Thank you for telling me that. So we have answered our question, what are the allele frequencies? Right, we've calculated out 43% and 57% respectively. We have looked at the genotype frequencies and calculated those out, 19%, 50%, 33% respectively. Okay. And that all came from just that little bit of information that 65 of 200 deer were white. All right, I think the best way to practice Hardy-Weinberg is to do it. So we're doing something a little bit different. We're actually doing the content check right now. So our content check is going to be an example of Hardy-Weinberg, very, very similar to what we just did. All right. So we have a population of butterflies. They are either brown or white. White, again, is the recessive allele, as white tends to be in nature anyways. Given this information, that 40% of the butterflies are white, I want you to tell me what percentage of butterflies are heterozygous and what is the frequency of homozygous dominant individuals. So you'll have to go through, just like we did, use both of the Hardy-Weinberg equations to come up with an answer. Now I'm going to give us 15 minutes in class to do this, right? I believe all of you can finish in 15 minutes, but if you need that extra time, right, I am not going to grade this assignment tonight. You will have the rest of the day if needed. But this is also a great time to ask me questions if you need any clarifications. Yes? You'll have to use both in order to calculate it out. Yeah, you can just use the Hardy-Weinberg equations.
I know. Oh wait. Q squared plus 2 plus.
Yes, and P plus Q equals 1. Yep.
And which one do you
Well, what do we know? We also know 40% of the butterflies are white. Yes.
Okay, so that leaves us with 60%.
Kind of. So 40% are white. Does that tell us the allele frequency or the genotype frequency being a white butterfly? Genotype. Yeah. So genotypes that have one allele or two. So we have to break it down and see how we want to adjust a single A. So I think you might have walked in after we talked about it, so I would work with Logan here to figure out how do we get there. Well, then look at the notes, and hopefully the notes will tell you.
I was listening, just pretty.
Great again. Will you be able to like?
I can read that. Yes.
Is it a campus? It's right after this.
Okay.
Just go on, and I can read the
Remind me your name. Yes.
My dad went here. So he came back to show me this and sell me on it.
Yes, yes. Do you want to come? Just like your dad.
Yeah, I do when I got here.
Did you get to see?
That's
Yes.
I'm going tonight or tomorrow.
Well, nice to have you in class. This class is like a broad overview of biology. It's the first one you would do if you're interested in biology. It covers everything from like atoms and molecules and biology to ecology, which is then after this, the class is kind of dissected too. Now we're only looking at organisms. Now we're only looking at genetics. Yeah. Nice to meet you. Let me know if you have questions.
So tonight.
About 40% of them are white. So I haven't given you the total population, but I've already given you the percentage. So you don't even need to know the population number in this case.
I'm so lost.
We had a question.
I'm looking at this in my folder.
Yeah.
Yeah. What do I do?
No.
I'm still looking to do like 40%.
So this means, so what does that count? Now, based on that, what it came to the first one was that.
Anybody really smart want to help? Anna, I need help. That's
You said two.
So let's, okay, wait, hold on, so B.
Okay.
So we're gonna say
And then you plug that into the P
positive or
Okay, then what's P?
giant.
How'd you get there? I'm more of like a visual learner.
No, you're right.
No.
What's what happens when you miss things like that?
I was playing OG Fortnite, you know? That is the total justifiable excuse.
So then well, hold on. Cause you need to do.
Alex, can you help me? Please? Pretty please?
No, but that's
I don't know how to do it.
So like.
No, I,
How about you come over here?
no, but I feel like it's like a teacher usually comes to the students.
So I feel like it's just
it's actually not. Because here, so here it says 35% are AA. So ours is 30.4%. 40%.
Yeah.
Right? So then we have to do the 0.3, so ours would be 0.4 to square, like.
Oh, right, it seems intimidating at first.
This actually
I know, that's why I'm trying to do it, so I then point to it, it's like going back.
Sorry, this is making sense in my head, so it's not. So the BB is 0.4. But we don't know which, we don't know how this is. So this one is going to the person for that. So the square root of.
What's 1 minus 1
Thank you.
Great work.
No, thank you.
Hey.
You're welcome. Said thank you 3 times.
That's great.
Alright, what do we get?
It's common sense. I've literally made everything like common sense.
I got it.
I didn't see them study on there.
Oh, round it a little bit.
Just why do I need, how do I spell that? Oh, wait, there's no.
So then for the.
We should have done this last night, like the responses person. What were you doing in the library? Exactly, it's not even doing this.
No, that's the percentage.
This is not what it is.
Cause see how she uses the.
I didn't know that was like a thing.
Is the square root.
Now I gotta get 0.37.
So,
Times
1%. Yeah.
What is 0.62?
Cause there's two square. So then we would do.
You're to add up all the individuals that were provided, there's one instance of.
So for B plus B prime equals 1.
That would be less than
I would say A.
instead of.
So then for the double B squared, double capital B squared.
Yes, yeah. When I hear you talk,
figure out.
Yes. All right. How's it going? That is correct.
Oh, wait, how many are there in the population? Oh. That's 200 again.
I think we had our hand up, right?
I messed up that.
Oh, okay. I'm gonna come back there and then I'll jump over here. I think the middle number you forgot.
Yes.
So I just take two. Oh, that's. Actually, okay, that's it.
Then that's it, yeah, it'll work and you'll apply it with
Is it 4.662?
Yeah, so then how do we find this? Is it just a single x? Okay, so we got 0.63, and then how do we get big D? Yes, you got that.
Yeah.
Yes, that's a good check, because there is something off there. So, we have E squared equals, we have 135, my answers, I rounded it, put 1/4, same, works.
I understand wanting me
No, I'm fine.
What if we split it down into chunks? Like, it's, I'm wondering if you're doing it. So what if we did 0.135 times 0.632 and then multiply that by 2? I don't get it by 0.135.
I'm so lost.
Yeah. Yeah, okay, that's
I know.
I normally say equal to dominant if it was this way.
But yeah,
So then.
Is it that easy?
That's
I don't want to go with yeah, that would be a marvel. What is and then squared equals
my
We have your answers, you just have to interpret which one goes to one, number one and number two.
And then my grandparents are coming tomorrow, and I'm going out to lunch with them too.
All right, that 15 minutes flew by. Question?
Question now that my answers are right.
Yes. You have a question? Yes.
I know this like nothing else. That might help me better.
Yeah. All right, so 15 minutes, that was fast, right? I thought that would feel like a lot longer. Are there any questions on how to do this? All right, there is guaranteed to be a question like this on the final exam. I am going to bring in calculators for the final exam. The best way to study this is to practice example problems. I have a whole list of example problems up on Moodle that you can go through. There's also an example problem at the end of this slideshow that'll tell you the questions and then the answers on the next slide. Right, so plenty of opportunities for practice. My other class asked me, will I give you the equations for Weinberg? I think I'll give you those equations. Right, I won't have you memorize P plus Q equals 1, though you should, and in upper class classes you will need to. You continue in the biology path. But for now, I'm really concerned with how you apply it to a situation. Can you apply it correctly? So I would give you those equations. All right, any other questions? All right, well, we are going to continue to a new topic then. Right. We're going to talk about speciation. And yes, before you leave today, make sure you turn in that content check if you did it on paper to the front. So I like talking about speciation. I think it's fun and interesting just to see the variety that exists in nature. All right, so we're going to talk about what is a species, how do we distinguish species apart, right? So first of all, what is a species? How do I identify a species, right? Well, there's some terminology that we have to understand before we can dive into that question. Just like whenever we start a new topic, we have a little bit of terminology. So we could say microevolution, right, is the evolution on a small scale, right? It's that change in allele frequencies in a population over time, right? It's how we get a population of brown mice, right, that becomes, over successive generations, a population of white mice. Right? We're changing those allele frequencies. Whereas macroevolution, we're talking about evolutionary changes that lead to a new species, right? Not just a different fur color, but a completely new species. Now, macroevolution is typically the result of many, many, many generations of microevolutions within a population, right? So macroevolution, think big time scale. Now, a species is what might be the result of macroevolution, is a different species. So how might we define a species? Very broadly, we would say a species is a group of organisms that share a distinctive set of attributes. That is a very broad definition, right? What attributes, right? How much do they share? We are going to see through the next few slides exactly how this definition can be torn apart a little bit and how do we define a species. But very broadly, we would say organisms that share a set of attributes. And then the process of making a new species is speciation. Right? And again, this happens over many, many generations. It's the accumulation of many microevolutionary changes. And we get that new species. So how do we go from, say, a wolf to all these different breeds of dogs? Those are examples of microevolution, right? Same species, just different frequencies of certain alleles. Versus how do we go from a fish to an amphibian in the evolutionary time scale of the Earth, right? That would be macroevolution. Completely new species has arisen as a result of many, many, many microevolutionary changes. Right, so microevolution, change in allele frequencies. Macroevolution, right, where we get a completely new species arise in the evolutionary chain of things. Species is organisms that have the common attributes. And speciation is the process of making that species. I know there's a lot of text on this slide. All right. So how do we define a species? So if we need to distinguish out a species, there's a few different ways that we can do so. First of all, the population we are looking at has to be a sexually reproducing species. They cannot reproduce asexually. Right? If they reproduce asexually, they're just clones of one another. Rather, they are reproducing sexually, so they can interbreed with each other, but not with other species. Right? So that's the first defining characteristic in determining, is it a species? Right? But there's also characteristics that we can use, such as morphological traits. So what do they look like? Ability to interbreed, right? Maybe they can breed, but they are isolated in some way, so they physically cannot breed with one another. Molecular features, so we're looking at the genetic code, as well as ecological features. And this varies on the species in question. Which one are you going to focus on when determining, is it a separate species? Are we going to focus on those morphological traits? Or are we going to focus on the DNA and the genetic code? Completely varies depending on the organism in question. So we're going to go through all of these different characteristics to see some examples of each. And we're going to start with those morphological characteristics. Right? So that's the physical characteristic of an organism. What do you see? Right? What is the body shape? What is the color of that organism? Right? And so here I have an example of some frogs. And I want you to tell me, with a show of fingers, 1 through 4, how many species do you see? I see some 2s, I see 1s, 3s. Yeah, it's kind of hard to tell, but maybe you're basing it on how they look. Those top 2 frogs are actually 2 of the same species. And the bottom 2 frogs are frogs of a different species. So there are 3 species shown on this image. Right? So this is where using morphological traits can be tricky. All right? There are many drawbacks to this. While the advantage might be it is really easy just to look at a population of frogs and see which ones look different from each other, right? There are these drawbacks. So first of all, how many traits are you going to consider, right? Are they just different in the color of their skin, right? Blue versus yellow, right? Or are they also different in their body shape? Maybe their head shape is totally different. Maybe they have webbed versus unwebbed feet, right? How many of those traits are you going to consider that have to be different for it to be a different species? Right? Traits, they're also continuous. Right? Some traits might be as simple as blue and yellow, but there might be a lot of traits that are in between, or could be a spectrum of colors. I know we talked about corn when we talked about polygenic genes, where multiple genes influence one trait. Right? So the color of corn can vary from very distinctively red to only white and every variation in between. Right? So traits can vary. How different do those traits have to be from one another that you're physically seeing for them to be considered different? Right? So what degree of dissimilarity are you going to use? So these bottom 2 frogs, right, they are different species, but they look very similar. maybe if we were looking at them and said, oh, this one on the right looks like it's a brighter green than the one on the left, right? Is that enough of a dissimilarity to consider them different species? It totally depends. Depends on the species we're looking at. Right? So members of the same species can look very different. Right? So what if, we had aliens visit us from outer space and took a look at the human population, right? Just based on how we look, do you think they would classify us all as the same species? It's hard to tell. We all look very different, but we do have common characteristics, right? We all have 2 legs, 2 arms, a head, right? A torso, right? But we all look very different. But there can be different species that look really similar to each other, right? Take these frogs, for example, right? They look really similar to each other, but they are in fact different, right? So there's a lot of different drawbacks, pros and cons to using just morphological features to determine if they are different species or not. All right, another type of mechanism that could be employed is reproductive isolation, right? Can a population of 2 different species interbreed with one another, right? Or maybe they're populations of what looks like the same species. Can they interbreed with one another, right? So again, this might be simplistic in that we could just observe a population and say, well, looks like population A is having offspring with population B. They're interbreeding. We're going to assume they're the same species. But again, there are drawbacks to that, right? It might be difficult to determine in nature, right? Some organisms, very different breeding patterns, right? Think about, would it be easy to see if like a worm is breeding with another population of worms, right? A, they breed very differently, right? And B, they might breed underground where you physically can't see them, right? There might be difficulties in determining this. Maybe populations, they could physically interbreed, right? So if you put an individual from population A next to population B, they could make offspring, right? But maybe they just don't for whatever reason, right? We might not know the reason. Maybe it could be that they're isolated by some geographic isolation, right? Maybe there's this river in between 2 populations, and even though they look like they're the same species, they're never going to be able to breed with one another. Thus, we might consider them different species based on that fact alone. And finally, we can't apply this to asexual species, right? Species that basically clone themselves. And we can't apply it to the fossil record, right? If species are extinct, we have no idea how they bred with one another, if they bred with one another, right? So we can't apply this to the fossil record. Right, now normally what this is used for is to distinguish closely related species of modern plants. Plants and animals that might look very similar to each other, we might assume they are a single species, but for whatever reason, they do not have the ability to reproduce with one another. Right? The pollen from one plant might be incompatible with the flower of another plant. Right? So today, that's kind of how we apply this mechanism. All right, so we have morphological traits. We have reproductive isolation. We can also have molecular features. All right, so this is where we're looking at the genetic code. All right, how different is the genetic code between 2 different species? Now, often this is applied when species look really similar to each other and act really similar to each other, and we can't identify it by observation alone. So bacteria, algae, cyanobacteria, these are good examples of species that we can only tell them as different species because of their genetic code. Right? So we're looking at the DNA sequences within their genes. Right? So is their DNA sequence the same? We're looking at the order of the genes. So remember, the location that a gene occurs on a chromosome is called a locus. So between species A and species B, is that gene always going to be on the same locus? Same with chromosome structure, right? Do the chromosomes physically look the same? Are they the same length? Do they have the centromere in the same spot? Same with chromosome number. All right, humans, we have 23 sets of chromosomes. Other organisms can have thousands of sets of chromosomes. Now this might seem straightforward, and you might think, why doesn't everybody do this? Well, the drawback is, where do we draw the line? How different in genetic code do species have to be for them to be considered different? If they differ by 1%, is that enough? What about 5%? 20%? Right? Where do we draw that line? It's very difficult to tell sometimes. And like I said, this is very commonly done for organisms that look and behave really closely to one another, like algae. In grad school, I had some friends, and this is what they did in their lab. They looked at algae, they determined the molecular sequences, and they identified new species simply based on the genetic code. All right, the last way that we can look at species is ecological factors. Right? So factors relating to how an organism uses its habitat. All right, so if we were to look at an example of bacteria, right, many bacterial species have been classified as being different based on how they use their environment, or which environments they are found in. Right? Maybe a species of bacteria is only found living on trees, and another species of bacteria is only found living on rocks. Right? Even if they look the same, act the same, reproduce the same, they're inhabiting two completely different environmental areas. We might consider them different just based on this fact alone. Same for these warblers up here, right? Even though they look the same, they're using their environment in a different way in how they forage for food, right? Maybe some of them only forage for bugs on the ground versus bugs on the tree. We might consider them different based on that alone. Now again, drawbacks, right? Sometimes, especially when we're looking at bacteria, they can display very similar growth characteristics, right? Even if the species are completely different, right? So maybe we have species A and B, right? They are inhabiting the same area, they have different growth patterns, but they might in fact be the same species. Or maybe they are different species. It can be so hard to tell sometimes, right? So pros and cons to each one of those different factors when trying to determine what is a species. So what I want us to do is take a few minutes here and try to identify what are the pros and cons of each. So my next slide is it's time for us to step back, but I know there's a lot of text. I see a lot of people writing. All right, I want you to tell me the advantages and disadvantages of each of these types of classifications.
I guess just
Reproductive isolation.
Yes, I think you can start on it. Right. I think you can start on it. I think it's a lot of
It's hard to tell.
I mean, tell on a
Yeah, you sort this pretty well.
Disadvantages is sometimes there are like barriers that happen to depending. I don't know. The advantage of I guess you can kind of see it.
Like they're just getting more disadvantaged.
Like most of them are not really in the
But I guess when you really want to like figure out what you want to do.
I don't know what they have are small to like you can disadvantage is that it's difficult to determine. Well, interbreeding. But it's just like, I just don't like yeah.
It's a lot easier to tell.
Yeah, like how accurate this means.
Yeah, this is a thing where options are simple.
Yeah.
Yeah, so all the kind of drawbacks on here are just disadvantages. Yeah, just like disadvantages.
All right, let's talk about these advantages and disadvantages. Morphological traits. What's the advantage of just looking at morphological traits to determine a species?
It's pretty easy to tell.
Yeah, it can be easy, right? You're just physically looking at two species and seeing, do they look different? Right, so easy to see, but what's a drawback there?
They could look very similar.
Yeah, they could look very different or very similar, right? And they could be different species, right? So think about those frogs. They look the same, but they were different species. Or reproductive isolation. Why is this an advantage of just using this method? If you were a scientist, how would you observe this?
I think it just like provides one species successfully from like interbreeding with others.
Yeah, so we're talking about can they breed. So if you're a scientist, a population scientist, going out and observing a population, you're just physically going to look, right? Is population A breeding with population B? It's pretty easy, right? They look similar, but do not interbreed. We can call them a different species. But why is there a drawback to this? You can't use it for everything, right? Maybe it's just hard to tell in nature. Maybe that they actually do have the ability to interbreed, right? Their genetics match, but they choose not to for whatever reason it is. Molecular features, what are the advantages?
Pretty surefire way to tell.
Yeah, you're looking at data. You're looking at genetic code. It can be easy to detect if a genetic code is different, right? But what's the drawback?
It's you don't know where to draw the line most of the time.
Yeah, where do we draw the line? How dissimilar do they have to be for them to be a different species, right? All right, last one, ecological features. What is an advantage here?
Okay.
What makes this one a good one to use when determining what's a species?
Things that live in the same location or different locations that look the same. I don't really know what that's called though.
Yeah, maybe things that use parts of the environment differently, organisms, right? If they use different habitats for food, we could consider the species different from each other. Maybe they use different habitats for nesting, right? We could consider them different. But the drawback is, right, many species have a lot of variation. It's just hard to see, like, are they actually using completely different habitats, right? It's just not as straightforward. All right. I'll pause for a couple seconds so you can finish I don't know what I did there. Finish writing things down. So in defining a species, it's not as straightforward as just saying that they have similar or dissimilar ways that they look, right? There's lots of nuances here. All right, so by far the large prevailing concept in biology that most biologists use when considering different species is the biological species concept, right? There are many ways to define a species. We just saw the different ways we could define a species, right? But the biological species concept focuses on reproductive isolation, right? It says a species is a group if they have the potential to interbreed with one another, if they can produce viable offspring, and that they're not able to breed with other species, right? This is the most common theory we have for a species concept. We're mostly basing it on, can they reproduce with one another, right? There are other concepts out there that focus more on the genetics, that focus more on the morphologies, right? There are other species concepts. This just happens to be the most common. Because it's the most common, that's the one we are going to focus on, right? So we're going to focus on the biological species concept, which really has to deal with reproductive isolation, right? Can species interbreed? If they do interbreed, can they produce offspring? If they produce offspring, is that offspring viable? So when we're thinking about the biological species concept, we have to elaborate on one of those characteristics we already talked about, which is reproductive isolation, right? Mechanisms that prevent interbreeding, right? And again, we have this nice image here. We have two populations of deer. They're isolated by this river, though this image always makes me laugh because this river is skinnier than the deer. They could just jump over in this image. But we're assuming there's a barrier, right? They can't interact with each other on either side of the river. Over time, because they've been isolated, they've developed different changes, right? This deer tends to be more of a darker color. This population is a lighter color, right? Those allele frequencies are different because they've been isolated for so long, right? So it's consequences of these genetic changes that are happening over time because they've been isolated, and this can be how new species arise, right? So we're going to focus on reproductive isolation and the mechanisms by which we can isolate individuals, right? This is one of the ways new species can arise, the one that the biological species concept focuses on. There's a lot of different mechanisms by which this can happen. So far, we really only touched about habitat isolation, right, where they're physically separated from each other by a river, a mountain stream, some physical geographic barrier, right? That is an example of a prezygotic isolating mechanism, meaning it prevents the formation of a zygote, right? Gametes from each individual physically will never be fertilized, right? So I have this nice flow chart here, right? Our gametes are represented by colors here, the red gamete and the yellow gamete. And there's all these different mechanisms by which prevent those gametes from ever meeting, right? We really talked about habitat isolation, but we're going to go on to elaborate on other forms like temporal or behavioral isolation. But basically anything that prevents two gametes from fusing together to form a zygote would be prezygotic isolating mechanisms. And on the other hand, we have postzygotic mechanisms, where maybe the gametes actually fuse together and create a zygote, but the gamete or the zygote either doesn't survive, maybe it survives and produces offspring, but that offspring is sterile, or maybe the offspring isn't sterile, it can reproduce on its own, but there's just so many genetic disadvantages that that population is going to die out, right? Something about it is not viable after this zygote has been formed.
So it's, sorry, it's like when, like mules.
Exactly. Yeah. So I'm actually going to stop here because this is where my last class stopped, but we'll go on to talk about examples of each of these. Mules is a good example of a post-zygotic isolating mechanism. All right, as a reminder, there are a few homework assignments coming up next week. Keep these on your radar. The last slide?
Yeah, I didn't get down that second right there.
Yes. All right, and if you have that content check, go ahead and turn it in to the front. Do you know where you're headed to next?
Yeah, it's just a lot.
Perfect. Yes, not a problem. Okay.
No, you got third. I'm so far.
Oh, let me grab my cheat sheet.
Study nothing. Study something on the ground. What's the grind?
This, I think we just didn't square it. I think if you take .316 and square it, you'll get the correct answer. I'll do it in like an hour. Perfect.
You didn't do the contact check? I don't know.
Yeah. Technically, it's due in an hour. I am not going to grade it today, but I can't guarantee I won't grade it over the weekend. Yes, that is fine. Yes.
Actually not that bad.
Oh, I said my office.
My finger just started bleeding.
Oh.
And it was my middle finger too, and I was like, what?
Oh no! We'll like wash it off with some soap and water. Yeah.
Wait.
Got another one?
Why, there's more over here.
That is your content checks, yes.
Yeah, it says your name.
Test corrections are.
Thank you. Have a fantastic weekend.
Bye.
Here's my content trick.
Perfect. Let's see.
Oh my god,
I
Megan,
go.
you should come to the path with me so that I can show you the. Don't, I have a path. Okay, well then. I purposely have not read it last or something. Okay, well, I'll go to the calf and then you can go pack and then I can show you my
I'm swimming a
package.
mile. I'm swimming 2,800 yards just in races alone.
I heard it's like what, a 2-day event?
Yeah, it's 2 days. We have 3 sessions of swimming.
Wow.
I've been able to swim for 4 weeks, and he was like, "Yeah, she's ready to swim the mile."
Oh no. Well, good luck.
Thank you.
Yes.
Have a good day.
Come on in. I'm just packing up.
Sorry.
You're quite fine.